All right, good morning, everyone. Hope you had some good sleep. Today we'll talk a little bit about what happened to your brain when you slept and why that's important for abstraction and intelligence. So last time we covered all of 6 slides. So let's see if we can, I don't want to go too fast. Keep the questions coming, but so today we are going to move into a little bit more than just background. So let's get a little started on that whole thing about what we can do with technologies and so on to understand neural circuits, because measurement is a key challenge. So it turns out that there is this tiny part of the brain called hippocampus. You heard about that from Ida yesterday. Which does all sorts of funny things, and the funny thing is that size part of the brain about that size of your thumb. So brain about that big, hippocampus about the size of 2 thumbs. 1/3 of all the neurological diseases, learning, memory, recollection, ADHD, Alzheimer's, they all are in that tiny part of the brain. The brain, as we discussed, has 100 billion neurons, which is not so impressive, not that exciting, but it has 100 trillion connections overall. And that's a circuit that people have mapped out over the past 60 years. So this is the retina right there. These are retinal ganglion cells which send the output. This is, some people call retina itself as part of the brain, rightly so. But if you go to the central nervous system, there's this big fat cable called optic nerve that goes from your eye all the way to the central part of your brain called thalamus. From there, it goes all the way back to primary visual cortex, where you see these Gabor patches and lots of cool stuff. Then it goes into this explosion of all sorts of brain regions. We do all sorts of funky stuff. Some parts care for the contour or the shape or texture. Some care for the speed at which something is moving, regardless of what the object is. So the brain breaks it down into all kinds of components that you are not aware of. When you're looking at this scene, your primary visual cortex only is looking at what part, where is the edge. It only cares for these little edges in my eyebrows, doesn't see the rest. V4, on the other hand, sees for these little textures and so on, doesn't see the edges. This part, on the other hand, cares for when something is moving, doesn't care for what really is, more or less. All this goes on, and this is only the visual cortical circuit. And then it starts to converge. It starts to converge, and all these areas converge onto this tiny area called EC. Stands for entorhinal cortex. Ida mentioned yesterday, it has these funky sorts of cells called grid cells. And then it ends up at this other area called HC. That is hippocampus, about that big. And it turns out that plasticity, which is one major form of learning that we think how the brain learns, is sealed in this part early on, like visual cortex, when I mentioned last time. It's all mostly done in the first 3 months, 3 years, depending on the animal. But as you start growing, there is some gradient of plasticity, and this part, hippocampus, remains plastic even in us. And if that plasticity in hippocampus is somewhat messed up, you are thoroughly lost. Yes, please.
I'm just, how many, in this picture, how many neurons and synapses are there? Like, of the 100 billion, how many are we seeing?
It depends on the species. I'm going to be talking about rats, but for us, visual cortex takes up about, this is the only cortex, so I don't want to talk about the rest of the brain. This is just a picture of, it takes about 1/3 of the neocortex. There are lots of neurons now. I don't want to do the math on the fly right now. I haven't thought, because half of the brain is in cerebellum, more or less, which is at the back, so you have to take out that half and so on. I would say this will be roughly ballpark 10 billion, roughly, but I haven't done the math, in that range. And then here is a tiny part. And this is not the only place everything converges. There are other areas too, which I won't talk about. Some convergence starts to happen, so there's this explosion from your retina to all sorts of regions, all kinds of specialists, if you wish. They do their specific things. They don't learn too much in the adults, they still learn. And then what starts to happen is that there are 2 points at which this starts to converge. One is the area called the IT cortex. And IT cortex is somewhere here, deep down. That's the one that's involved in recognizing objects, recognizing faces in an invariant fashion. So that's one direction that people study a lot. but that's somewhere here. Another place where things converge is the prefrontal cortex, which in humans is proportionately the largest compared to the rest of the brain. So people think that's where, so if you take the size of the prefrontal cortex, divide by size of the rest of the brain, rather than just the size of the brain, that's much bigger. And that's where people think decisions are being made, such as your so-called lizard part of the brain. There is no really such thing. Says food, eat. The prefrontal cortex says, no, wait a little. Wait for this guy to have some. So that's the prefrontal, yes.
What is the technology to produce such a diagram?
The technology, remarkably, that produced this was remarkably simple. Microelectrode. So what found was that people found you want to measure the activity of neurons. If you want to measure activity of neuron in behaving animal, you can't patch it because neuron dies very quickly and you need to anesthetize the animal to patch, so that doesn't work. So around 60, 80 years ago, people found if you take tungsten, it has high impedance, it doesn't get corroded, and they made a microwire. Tungsten is annealable and it's pretty hard. So you get a tungsten tip, which is 5 micrometer, you stick it in the brain and it remains reasonably stable. And because of high impedance, it can sneak up very, very close to neuron. Neuron cell bodies where the spikes are seen. That story changes in the future, which I'll come to later. And now you sneak up very, very close to the neuron. Its subthreshold membrane fluctuation you can't pick up because you're outside. It's 10 millivolt fluctuation you can't pick up. There are so many other neurons. But the spikes that are 100 millivolt, that 1 over r square will pick up as some 100 microvolt fluctuation. And when electronics got better, people could pick it up. Yeah.
You cannot analyze 100 billion.
You don't do 100 billion at a time. So what people did is 2 sets of techniques, which I can go into some other time, which means never, which is to stick the electrode in some part, first of all. And before you stick that electrode in the part, people say, all right, let me see what happens to a guy or a girl or a rat, usually a rat. You can't do that in humans. So let me take out hippocampus, just suck it out. And the way they kill it is to literally, they used to do it, it's called aspiration. A nice word for, so hope for the brain doesn't go in your mouth. You stick it in there, you suck out that part of the brain. That's how technology, human beings are quite inventive. Things got better these days, but that's how it started. And they say, I take it out and let's see. Seal up the brain and let the guy go around and see what the deficit is. That's called lesions. Or naturally, they know that somebody has some deficit in hippocampus, they had Alzheimer's, autism, there's some weird thing going on, and now we see what was going wrong. So based on that, people say, all right, if I want to understand if somebody has vision deficits, I better go somewhere here. But like Oliver Sacks wrote this book called The Man Who Mistook His Wife for a Hat. If you haven't read it, I encourage you to read it. Did this work right here at UCLA. So one for us. Where if you have a stroke or something in some of these further deeper areas, like this Mr. X who had a damage due to stroke, this part gets stroke, and now whenever he saw his wife, he thought it was a hat and tried to hang things on that and so on. He could see everything well. He could sketch a picture of his wife and he could see other things quite well, but just the wife part, it's a hat. That's the title of the book. So it does weird things. That's why I want to re-emphasize the claim I made, my one claim that thou shalt not introspect about the brain. You can introspect about the mind, but brain does very weird things. You had a question, yeah.
Just what do the edges actually represent? I mean, are they like parts of
those are basically axons.
With each other, or?
Those are axons.
But not individual ones. I mean, just huge.
No, it's just a block. Then it gets, this is like 40 years old. This has changed quite a bit, and people have now developed newer technologies where you can put kind of viral vectors, or even in human beings, you can put in an MRI scanner and something called diffusion tensor imaging or the blood flow and so on. Based on that, you can get much more nuanced picture, but this is still true. And these arrows are basically axons, and that is true. Axons is where the signal goes out of the neuron. And quite often, these arrows are not one directional. That's why you don't see a tip. So from here, it goes here, and it also goes back. And people debate quite a bit about how much feed forward versus feedback. Just please.
Going back to technology, can you actually see or get direct evidence for the axon, or is it just inferred from
axon, this anatomy, so solid. The fact that these projections exist based on anatomy, you take a brain slice, you image it and see where the axons are going, people do other things, take a brain slice, stimulate in one part, see if the other part is activated or not. So that's pretty solid. The question starts to get more tricky because every part of the brain gets multiple inputs. And what goes forward also invariably comes back. So then it starts to get tricky, and then the fact is within each brain region there is inhibition. So that can reverse the sign, and it can cancel things. So that starts to functional connectivity is harder. Yeah?
How much variation is there in this between people?
Not much. Remarkably stable. Remarkably stable. Now, it changes across species, but within a species, amazingly stable on a macroscopic level. Microscopic level, things can change. There was a hand in the back, yeah.
Yeah, completely naive question. But like, if I just looked at the brain, I would just think it's a lump of pretty homogeneous matter. So how do you identify different subregions? Is it based on like connectivity patterns, like?
Yeah, that's right. So the blob looks like a blob because the nature decided they don't want us. They found us very dangerous, so it should be hidden from us. Don't let human beings anywhere near. But then human beings violated that. So then you can see based on a lot of things. For example, you can just take a patch of the brain and dump a stain. This was an interesting historical accident. A bunch of scientists wanted to see the neurons, and they found a dye called Nissl stain. Now, the cool thing about the dye was it was imperfect. It was so imperfect, it stained only 5% of neurons. But that was a great part, because if you stain every neuron, you'll see just another, instead of gray blob, you'll see a blue blob. Then people started to see this, and a great scientist called Ramon y Cajal in Spain, he could trace these things, and half of the circuit anatomically he traced off in, like, God knows, 120 years ago. So there is solid evidence. It's not by any means hazy or imprecise or something. It's just complicated. Yes?
So if you think of the edges as the axons.
By edges, they're just connections. Let's just call them connections.
Sure.
Yeah.
I don't know a lot about brains. I don't know
Sure,
a lot
sure.
about graphs, but I know about nodes and bonds. And so we said yesterday there's no action-reaction, but there is some information exchange.
Yes.
So there is some principle there.
Yes, this is sending information. It's sending activity. What it's sending is activity. A whole bunch of neurons, when they become active, that's when neurons fire spike. When the neurons fire spike, that signal travels down the axon. These are the axons. Now, what that other region does with it, that's a giant question. But what these axons send is activity, in particular action potentials. So action potentials travel down this, and then they have synapses, and they release chemical, and then a whole bunch of complicated stuff happens.
But something gets sent back sometimes?
That's why these axons are going this way as well as that way. So that's why these lines are just lines rather than just arrows. And the weights are different, highly debated. How much is feed forward? How much is feed back? I don't want to get into that now. All right? Everybody cool? So that's what happens. And then that's the deepest part. And most people, when they think of intelligence, they think of prefrontal cortex because that's bigger in human beings relatively than anything else. But I have a slightly different take. I believe anatomy actually says that compared to deep network, that is the output stage, and we must think of it as such. And also, it's highly plastic. So what does hippocampus do? That's number one. Has all these diseases, remarkably, none of them except epilepsy, which is largely solved but not entirely, we can't cure them. We can cure a lot of things, but this we can't, and that's important to keep in mind. And the way this thing is supposed to work from something that you saw, because when you have damage to hippocampus, as we learned yesterday from patient HM, discovered by this amazing scientist called Brenda Milner, which it's a travesty she never got a Nobel Prize. But she discovered that when there was this patient HM, when they took out his hippocampus, because hippocampus is also, as written here, has tons of epilepsy. And epilepsy is so intractable, it's like a bit of infection, as you wish, electrical infection. If you don't cure that epilepsy, the epileptic seizures start to spread, and then they go to important things like breathing, and the patient dies. So you want to take out the tissue so the electrical storm doesn't spread. When they took out hippocampus in this famous patient HM, he lived a pretty long and healthy life. But he had a weird problem. You can come to the room and you can have a conversation with him for like 5 minutes. It'll be a reasonably normal conversation. And then say, hey, hang on one sec, I want to go out and get water. You come back with your glass of water and say, hello, I'm Mayank. And you say, hi, nice to meet you. So, all memory lost. But the memory of what happened before his hippocampus was removed is still there. So people came up with this idea that the way hippocampus does this thing, back in the days, this is when the idea was proposed by David Marr, that back in the days, computers required a strong man, right? These days, computers require only strong fingers. Soon enough, that will also go. Needed a strong man. So in the daytime, the computers do all the stuff, and in the night, the strong man comes with a huge tape drive and sticks it in and takes the backup. So, aha, when we are behaving, when we are doing stuff, the hippocampus is learning everything. And when you go to sleep, now this learning network is gone. Your eyes are closed. You don't hear anything. You don't smell much, this and that. So that's when the hippocampus is taking the backup into the neocortex. That's the prevailing idea of how, and that's where memory, so memory is formed during behavior. Some information comes in, let's say through the eye, similar circuit for audition and other circuit, which we are not looking right now. All that information is learned by hippocampus, more or less like a scratch pad, which you are doing right now. You're taking notes furiously. And then you don't know really what really happened, but you write it down anyway. And then when you go to sleep, that hippocampus sends that information back to, say, visual cortex. That's what happened. And empty cortex, that's what really happened. And this other thing didn't really happen or not relevant. Some kind of memory consolidation. So it's a solid amount of evidence that that's roughly how all of this works. Yes, please.
So is the hippocampus where all like episodic and semantic and procedural memory is? Or?
Thank you. So that's the major wars have gone on for longest time. But people debate whether the memories in hippocampus are only spatial or only declarative or episodic. I try to go away from that. I'm a physicist. I live in the next door. I like to calculate very simple things to basically annoy everybody. So that's what we are going to do. So we are going to go away from this, but by the time we finish tomorrow, I promise, if there is an interest, by now I have made too many promises, so we'll take a poll. What should I talk about tomorrow? And then we can talk about that. So we have done some work to reconcile these things. Long story. So now the cool part is that it turns out, as I showed you, that whenever the animal is doing something, for example, in a rat, you put an electrode, the rat starts to walk around, the neuron fires, one neuron fires only when he's here, other neuron fires only when he's here, third fires only when he's here, and so on. And individual neurons in hippocampus, so in a room of this size, individual neurons have selectivity that's about this big. About 1/10 of the environment, more or less. Some standard deviation. Now with one neuron, I can localize myself with only that much accuracy, which is no good. I'm going to bump into things. If you take the matrix of activity of just 100 neurons, and hippocampus in rat, which is what I'm going to focus on, has like 500,000 neurons. So if you take the pattern of activity distributed across just 100 neurons, you can tell where the animal is with an accuracy of 0.5 centimeter. And by you, I mean a useless physicist like me, a theoretical physicist of that sort. I could do that very reliable. So there is actually something funny going on in hippocampus, that it has 500,000 neurons, but a dumb algorithm, just say measure the average activity of all the neurons in a window of 100 milliseconds. Compute the population vector, see where does it maximum overlap occurs. That is accurate up to 0.5 centimeter in a rat with just 100 neurons. Why do you need 500,000? That's not redundancy. That's a massive explosion. And the brain wants to save energy, as we agreed on one principle. The other weird thing is that you say, all right, for some reasons and or the other, redundancy, the hippocampus is 500,000. But if I were to take you out from this room, and you have more neurons in hippocampus than rat by about a factor of 5, if I were to tell you, tell me when I took you out, were you here or very here. Do you see that? It's half a centimeter. No chance. So you do not have so-called declarative or whatever access to the even like a microscopic capacity of hippocampus. What's going on there? How can it be? So I won't get into that. Another promise I won't keep. But if you want, we can get into that maybe depending on what people want. And that's how it's supposed to form memory, by this two-step process. One step, learn very rapidly using this amazing plasticity of all these areas. The largest amount of plasticity is in the hippocampus, synaptic plasticity. You make some activity, the synapse changes. We'll look at that soon enough. And people believe, as the Nobel Prize has been awarded, that these neurons encode space, and the reason we have deficits in episodic memory or declarative memory is because space is the context. And if I take out spatial context, then I don't know what memory is. That's the prevailing belief. Now, I don't happen to subscribe to that 100% because you may not really remember what happened in this room, but you remember the conversation. Tomorrow, if I were to ask you which seat were you sitting on yesterday, most likely you'll have no idea, and that's way more resolution than half a centimeter, but you'll have a much better idea of what was going on in this room. So I don't subscribe to that, and if there is time, I will tell you an alternate theory, and that's what happens during sleep. So let me actually get out of that now, and let me move on quickly to this rule. So Mr. Hebb, in 1949, so now we are switching gears. How does it all come about? So I'm going to attempt to be Kepler. So can I start making a simple mathematical theory, going to be majorly flawed, I'm going to ignore a massive amount of stuff, but it's 100% biological, with lots of things thrown away, but 100% biological. So many people attempted this, and Hebb said, how do neurons do anything? So he said, groups of neurons which tend to fire together form a cell assembly whose activity can persist after the triggering event and serves to represent it. So what that says briefly is there are a bunch of excitatory neurons, and if some stimulus comes in, some group of excitatory neurons will be activated. Then you turn off the stimulus, the memory of the stimulus will be maintained for some time by these guys talking to each other, kind of keeping it up, keeping it active. So that's roughly the idea. How it works and so on, this was a conjecture. But then Mr. Hebb made another proposition that thinking is the sequential activation of sets of cell assemblies. So there's one cell assembly for my sentence number one, other cell assembly for sentence number two, something like that to put it together, and that's what causes this. Then number three is the most important part. How does it get learned? So he claimed that when an axon of a cell A is near enough to excite cell B and repeatedly or persistently take part in firing it, something happens, like some growth process or metabolic change takes place in one or both cells such that A's efficacy as of the cell B's firing is increased. So this complicated sentence basically says if one neuron generated spike, it activated somehow, the other neuron doesn't know if this neuron made it spike or not, because every output neuron needs the input of at least 10 neurons, and it doesn't know who activated. All that it says is, if this neuron got activated and then the other neuron got activated, synapse from A to B should get stronger. And if that happens, and the other two axioms that he wrote down happens, everything will be fine. That was the idea. The question is, how does it work? What does biology do? These are nice ideas, but if you want to understand biology from somewhat physics style, we say, I want to know the biophysics of what happened. Yes, please.
When you think of brain as some parallel, quote-unquote, computer, is it a coarse grain or fine grain architecture? Basically, is neuron a significant processor?
Yes, definitely. In fact, I will show you. In fact, no, well, let me not. There is some coarse graining beyond a neuron, as I mentioned yesterday, called columnar architecture in neocortex. There is the coarse graining definitely about 10,000
It's
is...
not a simple device.
No, neuron is not a simple device. Neuron itself is fairly complex. Another promise I won't keep, we have done some research to see how the neuron, which is neuron cell body is only 20 micrometer, but these dendrites, they are long, way longer than necessary, right? Ideally, a neuron should be just a sphere, so the input, the synaptic input comes in, reaches the deciding whatever thing, decision-making, spike generation thing very quickly. But neurons, on the other hand, are one-dimensional creatures, or 1.2-dimensional creatures, very thin, long axons or dendrites going up to 1,000 micrometers. That's where lots of funny things happens. We can maybe come to that a little bit. Yes, please.
And what do we know about non-neurons being computational?
Yeah, a lot is known. In the brain, the number of neurons that are not neurons, in particular glial cells, are 10 times as many as the neurons. They are much smaller, so they don't occupy that much volume. It's known that glial cells play a lot of role in lots of things. It's harder to measure what happens in glial cell because they don't generate spikes. If you don't generate spikes, microelectrodes don't work, so you need newer technologies to figure out what happens there, which just started to get discovered. I'll come back to that if necessary. Maybe not, all right? So now we studied that synapse last time, that there is this vesicles are released when action potential arrives, that when the vesicle comes, fuses here, releases neurotransmitter, that neurotransmitter crosses this 20 nanometer gap, binds to the neuroreceptors, and once it binds to the neuroreceptor like a lock and key, it's like basically a tetrameric structure, so it just swings around like this, like this. That's right, it's a long structure, swings around like this, and individual ions start to flow. In particular, the excitated neurons conduct, now this is where it starts to get very cool. Somebody yesterday was talking about all sorts of very fast time scales. These individual channels start to conduct one sodium atom at a time, one at a time, exceedingly small number. And it's about that big, about half a micrometer or thick, less than that. And the flow rate is about 1,000 sodium atoms per millisecond. Pretty slow, very efficient. So that happens. So we summarize inputs arrive at dendrites, travel down to the axon. So this is where all the inputs from synapses arrive. It sums it up. Then there's like a neck-leg process here. So all the voltages, all the current that are coming on, V equals IR. Evolution figured out Ohm's law long ago to say that here if you make the neck shorter, then the voltage will shoot up for the same amount of current. And then it packed that region with voltage-gated sodium channels, which boost this thing and generate a spike. That spike, nature figured out Ohm's law, Kirchhoff's law to say need to be insulated, so put a bunch of fat. This fat comes from the glial cells, to your question. So that's a strong thing going on, and that goes here, and that dumps that stuff onto the, God knows, 10,000 other neurons, and that's how it works. And now let's focus onto this synapse. These presynaptic vesicles, there are two types, one that release glutamate, other that release GABA. When glutamate is released, it injects sodium or calcium ions. When GABA is released, it releases chloride ions. You don't have to know this. Maybe I should just skip it, otherwise it will confuse people. So let me just tell you very briefly the fundamental mechanism by which this kind of causal learning, cause and event, cause and effect is supposed to happen. So here's now presynaptic site. It releases some vesicles. On the postsynaptic site, you have this one kind of receptor called AMPA. It conducts sodium. And as the more sodium, sodium is 1 plus charge. So to postsynaptic spine, and neuron starts to get more and more positive. That's the standard operation. But in addition to that, there is this another kind of receptor called NMDA, N-methyl D-aspartame, another acid. Don't worry about the name. But it gets used quite a lot, so if you're serious about it, know the name. Now, here, as soon as glutamate comes in here, it just opens up, things start to travel, easy part. Here, it's really funky. There is one magnesium ion sitting there. And it doesn't let glutamate open this. It doesn't let it open. What it requires is, because magnesium ion is positive, it requires that this little thing, in order for it to go through conformational change and then let this go through, this magnesium ion has to be repelled by giant positive voltage. If a giant positive voltage is generated, this guy is kicked out. So it's now kicked out. And now, another thing, this little hole lets in calcium ions. This thing, so these holes are magical, right? This hole, by giant efficiency, only lets in sodium, not potassium, not chloride, not calcium, not backwards, mostly or largely. So highly selective machines. These are proteins. This guy, on the other hand, lets in only calcium ions. Only if two things happen. One, glutamate has to come, has to do that, but then on top of it, it has to get a giant depolarization to kick out the magnesium. And that magnesium is not free floating, it's hell. This guy holds on to his magnesium, doesn't want to lose it. So this gets kicked out, and now quickly calcium starts to go in, and then in a short time it closes, and the magnesium goes right back and says, oops, I don't want anybody to talk to me. Very funky machinery. Now what's the advantage? Why would the evolution? It's like when you study this thing, you say, Jesus Christ, are you kidding me? This happened by chance? Like, you get freak out a little bit. What the hell is all this? How such amazing selectivity and amazing like things, but that does something pretty amazing. So what now happens is yet another amazing machinery happens after that. There are a huge number of other proteins, which I'm not even showing you. What they do, they sense the flux of calcium. Imagine, right? They're like, how many AMPA receptors? You might think it must be macroscopic. In a synapse, there are like 50 AMPA receptors. Really small number. How many NMD receptors? Of that order. Now there are proteins here in this tiny volume of half a micrometer. They're calculating on a scale of millisecond how many ions came in. This is ridiculous. And they measure how many ions came in, and if the rate at the current, the calcium current, is very high, then this guy, through those proteins, sends messages to these AMPA receptors, hey guys, something good is happening, come on. More AMPA receptors get inserted. If, on the other hand, the amount of calcium current is not that high, forget zero, if it's zero, nothing happens, but if the amount of calcium current that's coming in, so remember, calcium current, not total current, in a millisecond, it's ridiculous. If that amount of current that's coming is not that high, it says, you guys, go. And these AMPA receptors start to move away from the synapse, go away somewhere. Crazy. Now what that does is that this becomes a coincidence detector on a scale of milliseconds. So if glutamate comes in and then this magnesium is kicked out, then you'll have the maximum flow rate of calcium. If, on the other hand, giant depolarization comes, magnesium goes out, and while the gate is closing, glutamate comes in, causality is violated because this synapse got depolarized first. This NMDA receptor got, magnesium block was removed first, and then glutamate came and says, nope, I don't like it. And this is on the scale of 10 milliseconds, way below cognition. Yes, please.
Is this the kind of common mechanism in pretty much any neurotransmitter?
Not any neuro, no. This is NMDA only. This is the most common type of plasticity. There are other forms of plasticity which are dependent on calcium. We are not going to go there. By far, if you're interested, I refer to a book by Hille that's already 30 years old, the fat series of things. Yes, please.
How far down evolution is this, exist?
Good question. I don't know. I wish I knew. If you don't mind, just ask GPT or somebody and tell me what happened. I would love to know. I don't know this. It's a good question. Yeah. It's hard to do this evolution part, but I don't know. Somebody must have looked at it. I don't know. Yeah.
I guess that was kind of the thing I was slightly in as well.
Yeah.
I feel like a path dependency in these things where, like with silicon, for example, everyone now makes it the same way.
Yeah.
There's a huge investment in the machinery that goes into that.
Yeah.
Is the same thing true? Like you essentially like a neurotransmitter is the kind of, and then you just
Good question.
all the way through because
Really good question. Please help me. Look it up and tell me later on what the answer is, or tell all of us. I don't know the answer. I wish I knew, but this is the cool part. There's a lot going on here, and one can ask really good questions. So, very simple. Two types of synapses, AMPA and MDA. Calcium influx on MDA is primarily responsible for induction of change in the AMPA receptors, so net strength of synapse. Large depolarizations such as, so if that depolarization, you can say how much? The amount of depolarization needed is of the order of 50 millivolt. If it's less than that, things go haywire. Exact number. As it happens, 50 millivolt or higher is exactly the size of action potential, about 100 millivolt. If that giant depolarization happens, the magnesium gets blocked out. Otherwise, things go wrong. And then low amount of calcium, no change, and intermediate amount of calcium, then make the synapse weaker. So that's roughly many years of work people have done. So let me skip this part because we are running out of time about how calcium does this and so on. And let me just come to this story. So this is, people found the Hebb's idea as biological implementation. So what is being shown here? This is from 96. So x-axis is time in minutes. Y-axis is the size of that synaptic potential. AMPA receptor mediated largely. Calcium doesn't do much of depolarization. AMPA receptor or sodium-based thing. So that's called excitatory postsynaptic potential. It's excitatory postsynaptic person on the other side, and potential means its voltage. They nicely normalize it, so we don't care how much the voltage is. It's of the order of 5 millivolts, and say initial value is 0. So you say here they isolated 2 neurons in the brain, not easy, found that they had a connection, and they give it a pulse and say, okay, up to this point, they said, okay, the synapse is reasonably stable. It has some variability, but on an average, when you average across many trials, it's stable. At this point, they did the manipulation to check Hebb's idea. So what they did is they activated the input neuron first and then the output neuron, because they had managed to impale both the input and output neuron in the brain, so pretty hard. So tiny one little pulse, input neuron, that input neuron called input because the synapse onto this, but not the reverse. Then activate this within 100 milliseconds. If they do it within 100 milliseconds for a little bit of time, then they stop doing anything, the synaptic strength goes up by about 20%. Huge error bars, and it remains for the remaining experiment. So the presynaptic neuron is active before the postsynaptic. The synaptic strength becomes higher and remains high for the remainder experiment. After that, the experiment finishes because nothing lives. That's why it stops at 50. People have checked this over a longer time. I'm not going to get into that. But if you now do the other thing, you do the same amount of activity, but you just do the slightly opposite. You inject the current in the output neuron, make it spike 1 millisecond, and then activate the input neuron before that. Then the synaptic strength actually goes down and remains low for a long time. Very cool. So then other set of people got involved. They said, let's vary the time scale. So let's take the final point of this thing and tell me if I vary the time here, it was plus 100, plus 20 millisecond and minus 20 millisecond. Instead of that, let me vary all sorts of things and let me give you the final amount. How much is the change in strength after half an hour if I did this thing once? And they found this learning rule. So x-axis, again, 2 neurons, activate the input neuron, then activate the output neuron. This is delta t is greater than 0. Notice here, if it is about 10 milliseconds, synaptic strength increase. Each circle is a different experiment, averaged over many times. Increase by about 100%, sometimes nothing, sometimes 100, a lot of variability, but on an average, here it increases massively. If you do it, input and then output by about 20 milliseconds, already drops quite a bit to 20%. And then if you go beyond that, nothing, it's just chance level. If you do the reverse, literally, now this is pretty crazy, right? This is like 5 millisecond difference. Here is input followed by output plus 5 millisecond, all of them positive. Here, massively negative. Again, if you go back to minus 20 millisecond, it's kind of even less, and you go beyond that, nothing. So this can be fitted by two exponentials. One exponential, which is this, other exponential, which is that. And it breaks the symmetry massively. And the time constant is of the order of 5, 10 milliseconds. That's it. Yes, please.
I'm wondering why you're saying exponential rather than like t to the minus 1.
Sorry?
Like, let's say that spike timing is t, like t to the minus 1 would also explain this, right?
No, it won't change the sign. It won't go this way and that way. You need two exponentials.
1 over t.
I mean,
1 over
negative
t.
t is.
1 over t will give you this. When you start to go on the other side, you'll have to start going this way. So people have looked at this thing carefully. The two exponentials is the best fit to this. But you can look at this. We can talk about that. So this is the learning rule that is overwhelmingly shown. So you have input followed by output at different time scales. You get this dependence. If you look at the reverse thing, you get this dependence. All right? So that's a fundamental biological rule. And all of that has to happen within this tiny window, about 20 milliseconds, in this very rapidly decaying function. So let me skip this part and come to how can it work. So now let's zoom out from the background and discuss what is the problem that we are interested in. Yes, please.
Does the neuron purely local? Does it only get information from its synapses, or does it also learn based on local information or something else?
Sorry, I couldn't hear you.
So does a neuron purely local? Does it only get information from its surrounding neurons, or does it get information from somewhere else?
First of all, I won't use the word information. I would just use axon, because information is who knows what neuron gets. It creates whatever information. But yes, vast majority of input that neuron gets in terms of ionic influx, that's what happens, is through this. Other stuff happens outside the synapses too, but I'm going to ignore that. It's harder to measure if it's outside the synapse, so let's just ignore that. Anything else? There was some other hand. Yeah.
Just so I'm clear, on the previous slide,
Yeah.
you're saying that if the timing is reversed, the signal or the connection strength gets weaker between them? It's not.
That's right. That's right. That's right. That's what shows that. That here the connection strength improves, but if you go backward, it goes down, and all of that has to happen within a short window. Yeah.
I just have the answer for you. So it looks like quick search Drosophila also seem to have.
There you go. So it might be an early evolution, might be an early invention. Yes?
This is kind of a follow-up on Paul's question. Like,
Yeah.
so if you look at, say, like 20 or 30 data points, and then you
Yeah.
decide on some function to fit it, like
Yeah.
exponential, but you could have picked a polynomial.
I know.
How do they do it?
It's actually, this was something, this is colloquially called two exponentials, but these are very hard experiments. There are very few experiments like this that are done. So we are not going to rely on what is the exact function. So just somehow commonly used.
Like you just pick something that kind of.
Pick something that seems to fit, but there's just not that many experiments. These are really hard experiments to do. So there's not enough data to
Data, right.
just not there to say, is it this or is it that function? Just not possible.
But also some things that easy to work with in other equations that it appears.
I see what you mean.
Yeah, so we can do lots of things, but this is, we can say whatever form you wish, but it's more or less this antisymmetric function. Okay? So now, yeah.
So we had a seizure or something like this, right? Which of course caused all kinds of
Yeah.
malfunctions of the system. But still people recover from that somehow, right? So is this effect temporary or
Yeah.
is there something else?
Again, I can keep repeating, a lot goes on, but that's one of the reasons why if seizures are not treated, things get worse. One of the reasons, and we did some work many years ago to show that if you don't treat seizures, then this thing will lead to this activity spreading and recruiting more and more neurons, and it can mess with the circuit function. You wrote a paper.
These kind of steady waves that you have, they reinforce
Yeah.
somehow.
It will reinforce some things and it will mess with some things. So very short version of this is that this kind of activity pattern will break something called attractors. It will link unnecessarily random memories together, and that's the problem, and there is a way to solve it. I can just give you a reference, it's on my webpage. But right now, let me not go there, if that's okay. There's a lot of cool stuff. So let me pose the problem that I'm interested in. The problem I'm interested in is to say, all right, at the level of behavior, something interesting happens. We are all interested in that. That happens because this neuron has some interesting receptive field properties. Some of them are more active, some are less active, some encode for vision, some encode for something else. So that's what's going on. And then all of that happens in turn because of some funky stuff that's going on at the level of individual neurons and synapses. And the question is, how do you go from one to another? How do you go across these? And there are two problems with it. The first problem is well advertised, that how can changes at the level of one synapse influence 10⁴ inputs on a given neuron, and then by the time you go to the whole brain to hopefully to generate behavior 10⁻14. So there's nice segregation of the scales. How do you bridge this? How do you go across these different scales? So that's one problem. And when I started to work in this, I realized there's actually another problem too, which people had not paid that much attention to, and that's a question of time. As we looked at it, individual action potential, rise time is 0.1 millisecond, learning precise within tens of milliseconds. Neural activity, typically when the rat walks around or does something, each neuron is active more or less in the scale of 1 second. And by the time you learn anything, such as this lecture, it's 10⁻³ seconds or even longer. So now you have 7 orders of magnitude difference in time scales as well. That's a nasty beast. And that's where these problems become different from problems in physics. In physics, you have stuff going on within the nucleus at a very fast scale, completely separated from what happens at the electron, because these guys, you don't have to worry about the exact thing that quarks do. They just hold proton and everything interacts. And then what happens at the level of individual electron and QED, you don't have to worry when the gas particles are interacting. The scales are coupled. The scales are coupled, both in space and time. So that's the problem we want to solve. How do we do that? So, and how does that the result of all this, and on top of it, everything is highly noisy, as far as we know, noisy, but lots of things govern it. How can behavior arise out of this? Can we do that? And what are the emergent properties? One thing is to say, how does behavior arise? But that's a top-down question. I want to ask a simpler question. If these weird things are going on, what will it do to the network? Because they're really funky things. They may do stuff that we haven't imagined. And we know already that thou shalt not introspect about the brain. It does something, and then some tiny fraction we are aware of. So I thought, all right, so let's turn this thing, and let me now turn from a biologist to a physicist. I'm going to annoy the hell out of all of you by making giant simplification. So bear with me. So we're going to say, all right, one thing all animals have to do, apart from recognizing this and that and reward and punishment and God knows what, is to do some causal association. There's something that happened now and then something that happened later on a short time scale. And then I can link it up to make giant chains. So x-axis is time in dimensionless units, y-axis is some probability of spiking, and we are going to say, all right, we are living in a one-dimensional world. So one stimulus just went by in this one-dimensional world, going at a constant velocity. And as a result of it, there was this neuron that was active, and it's a nice neuron. We're going to ignore the spikes. Imagine it's hundreds of neurons or thousands. It is some activation function, some bell-shaped curve. All right. And then there was another neuron which had yet another bell-shaped curve. And this is roughly the description in many parts. One neuron is active at this stimulus, x-axis is time, y-axis probability. What was the stimulus? Could be a physical rat moving around in a place cell, or it could be a bar of light moving in front of you. Or it could be any bunch of sounds that coming at you. It could be anything. We're going to make it precise in a second. And now, in this simple network, because this neuron number 1, or this neuron is active before that, that NMDA-dependent plasticity will make this synapse bigger. So the next time after this is induced, when this neuron is active, it will make this neuron active just a little earlier. And that's how this Hebbian learning rule or NMDA-dependent learning rule will create tiny little arrow based on experience. So the reverse arrow won't grow, but this arrow will grow. reverse synapse actually will go weaker, so that will make the chain grow in this direction. That's the idea. Everybody cool with it? So that can solve lots of problems. So now, how does this work in a real network? So we measured the activity of many neurons. We want to summarize their property. These are the experiments I did. So we put a bunch of electrodes, not one or two, but lots of them, that the rat run back and forth on this track. And now we want to separate the reward location. So then you let him walk around on this track, so rewards are right next to each other. Put a partition, so he's going there. So the reward either is next to each other or far away. These are individual cells. There is one neuron, which later on I color-coded, which is active here. This other neuron active here. The third green neuron active here, fourth here, fifth here, sixth. So we summarize based on this, is neurons are active at specific locations in the maze. And when the rat goes out of this location, its firing rate drops to 0. This neuron is active here. There are practically no blue spikes here. This yellow neuron is active here, practically no yellow spikes here, and so on. So there is very low noise in terms of background activity. What I haven't shown you here is that these neurons are also direction selective. So this green neuron fires only when the rat goes here. The blue fires only when the rat goes here. The red fires only here, and so on and so forth. Majority of the neurons care, even on this tiny track. The track is only this wide, so rat barely fits in. So I don't have to worry about this time he was going here and that time he was going there. So I made practically a one-dimensional maze. And then these inhibitor neurons, 10% of them, they fire everywhere, so I don't care. I mean, they have some modulation, but we're going to make a simple story, so we're going to forget that. And as a result of it, this is a sparse code. So this neuron is active in less than 1/3 of the environment. Less than half, so it's a sparse code. And it's a directional code. So let me actually skip this. Let me skip this part and come to a simple theory. All right, so let's make a theory. Say, all right, I have one part in hippocampus has a name called CA1, doesn't matter. The standard theory that people had made before, the standard theory had recurrent networks. The recurrent that is found is only 5%, so I don't rely on recurrent network. But what I do know is that this one single neuron gets input from 10,000 neurons. And what is also known is that this one particular part of hippocampus has input from this other part, which has also spatial selectivity. Nice little place cell, they have all kinds of shapes, we're going to ignore it. So in this one-dimensional maze, let me start the simulation, all right? So now instead of going for one single neuron, we're going for more realistic circuit. So this is biologically accurate circuit. There is this part of hippocampus called CA1 that gets input from this other part called CA3. The same circuit works in other parts circuit. True, this could be primary visual cortex, and this is thalamus. This could be thalamus, and this could be retina, and some stimulus is moving by. So this part, this kind of a circuit, one neuron receives a bunch of input from the stage before that is true in all parts of the brain. The very general circuit, we are just going to be solving it for one simple thing. All right? I see people zoning out. Is this complicated? Should I slow down? All right, good. So let's just turn this thing. Let's do a simulation. So there's a stimulus that goes by, has some shape, which I can show you equations later on, but I'll run out of it. Some cosine shape bump. Different neuron is active as the rat goes by. So the next 1 minute is crucial. So pay attention, ask questions. If the next 1 minute is obvious, everything will become obvious and things will work. So now, what happens? So let me say I now took a picture of all the neurons' activity in one trial. So at a given moment, in about 1 second, first there is this neuron active, then there is that neuron, then there is that neuron, then there is that neuron. They're not next to each other in biological space. They could be anywhere. These are not all the neurons. These are a subset of neurons. So since the stimulus is one-dimensional, I can always arrange the active neurons in a one-dimensional sequence. Even random numbers I can arrange in ascending order. So easy. Now, some of these neurons have synapses onto this output neuron. Some others don't have. Those synapses are there, but they are very weak, so we can ignore them. We know that an output neuron will be active only if it gets input from at least 10 neurons simultaneously. Otherwise, the amplitude of the spikes don't match up. So then by definition, the neurons that are active and have a synapse onto this neuron, they must be next to each other. Because if one active neuron was here, another that was here, they will not add up. This will not create the output neuron to spike. So this is true by construction. So this is the first simple statement. The fact that output neuron will be active only if roughly 10 or more input neurons are active simultaneously requires that group of neurons which have selectivity here, they are all activate, they are all projecting to this, and the projecting neurons are not distributed randomly. How does that come about? I'm not going to go into that now, maybe tomorrow. Yes, please.
So far we've talked about like spikes, so neurons are active when they spike, and now we have these Gaussians.
Yeah, right now I'm just simple. As I told you, I'm going to annoy you. No, they cannot be half active. There's something like half happens, but this is the average activity across, let's say, 10 trials. The same thing is more or less going to happen. Or this is the input from like 100 neurons. There is serious weakness with this model because I'm ignoring spikes. But if we get to that, I will tell you how we can get out of that. So right now, bear with me. I'm going to be a totally annoying physicist making an amazingly simple model. Yes, please.
You'd mentioned the 10 inputs required to spike.
Yeah.
Should I be taking this quite literally of like...
Yeah. It's not exact 10.
Yeah.
But actually, so by the time a synaptic potential arrives at the cell body, it's about half a millivolt at most. And the neuron is typically get minus 65. So in order for it to reach plus minus 55, then only spike can happen. Then otherwise there is no spike. So you need of that order of simultaneous input in a window of 5 milliseconds. That's right, because AMP receptors are open for 5 milliseconds. So you do the math, roughly 5-10 spikes of that order. It's not hundreds and it's not one.
Yeah.
So it's in the range of 10. Yes?
What happens if the object just moves very slowly?
We're not going to do this. I'm a physicist now. It's moving at uniform speed. Not doing it. We'll come to that later.
I move at uniform speed for just slowly.
Don't want to do it. I don't want to do it. Right? We want to be Keplers. Let's not go into becoming Newtons, right? You first to go through that. So I'm going to be annoyingly simple. I warned you. Later on, we'll see what the speed does. All right? So that's what it does. Yes, please.
I'm still trying to catch up. So the output neuron is active, so it means that 10 had needed to be activated.
That's the only reason the output neuron is active. Because unlike the experiment in which somebody was messing with them, here there's nobody activating them.
And you're just saying that those 10 have to come from the strong?
They must have some strength, some non-zero strength. Otherwise, even though they're overlapping, there's no output.
Okay.
Right? That's all I'm saying. That's exactly right. I'm saying something almost trivial. But it requires some steps to say that trivial, and then we'll go to something non-trivial. All right? Yes, this must happen whenever an output neuron is active. Some overlapping neurons must be active, and those overlapping neurons I can always arrange in ascending order. No matter how precise the fields are, there will be slight jitter between them. I can always do that. Really simple, right? Now comes the final observation, all right? And you'll say, my God, this is trivial also, but that's how math works, series of trivial statements. Now I draw a line through the center of the output neuron. When I draw a line through the center of the output neuron, something magical happens. By construction, all these neurons, on an average, are active before the output neuron. So the output neuron is also responding to this. So all these neurons are going to, on an average, not 100%, are active before the output neuron. All the other neurons to the center, to the right of this, they are active after the output neuron. By construction. I just broke the symmetry because I created the symmetry by hand. They can do all sorts of weird things, but this is true always. Right? Everybody cool with it? Now let me bring out at individual synapses that NMDA-dependent plasticity rule. What will happen to this synapse? Will it get stronger or weaker? Stronger. What about this synapse? Will it get stronger or weaker? Which one will get stronger more, this or that? First one, because the time difference is very short. You go here, it flips. This synapse will actually get punished like hell. this synapse will actually get punished slightly less. And these, nothing will happen. So as a result of this, everybody cool with this? If you want, I can go through this. This can be then, one can do equations and show that this works. So this is actual simulation. You put two exponentials, we can put different functions. I use two exponentials because back in the day, people wanted to do that. And then you put this thing, and all of a sudden now what happens is that these synapses got weaker, so this got pushed back. It turns out, you notice that in picture, the net improvement or the increase in strength was bigger than decrease. So total area under the curve goes up. And now you have symmetry breaking. Your receptive field, we started with a symmetric shape. It developed this asymmetric ramping shape. These synapses got strong. These got punished, so they became yellow. And blue is the final strength. And these, which were yellow, became slightly blue, and this became much stronger. This is it. Yeah?
So let's say if we keep running this experiment,
yeah.
So after curve would shift toward the left, right?
That's right, it'll keep going forever. But hey, I'm a physicist, I don't care. Right? Some other day. You're going to come to that, what happens when it keeps going backwards and so on, but this is just a tiny snapshot. In reality, it cannot keep going forever. Because remember, these arrows are not just going this way, eventually what goes around comes around too, beyond the limits of this model. Yeah?
Yes, I thought you were saying that in order for the one on the bottom, the CA1 to fire, there have to be about 10
That's right.
on the top. But,
Yeah.
but it seems like the ones on the top are causing the ones on the
That's
bottom.
right.
So they should all be in the past. But you're saying that
No.
any that are accidentally in
That's
the
right. That's right. Good point. When the rat is here, is getting input from this as well as this. But when the rat is here, is getting input from a smaller number of neurons. So that's why the activity curve is like that. Right? Because just it runs out of neurons. Just by chance. If I just
you're
do a
saying
random,
ones that are
this will
firing
happen.
too late are getting punished.
That's right. That's right. Anything else? Yeah.
So I can imagine how this forms these associations of if this, then that.
Yes.
But you can also have associations like orange is yellow or orange.
Those are discrete ones.
Is there, but you could imagine that this being done purposefully by some sort of a delay where...
Yes.
So is there any evidence for that happening? Or?
Not obvious to me. I don't know. I mean, I just know limited amount. It's possible somebody, actually, hang on one sec. There was an experiment by this person named Miyashita. He did this experiment long ago. He showed a bunch of images to a monkey, random images. And there was some random task, so no reward, this and that. And then what he found at the end of the experiment, that the neurons that were reacting to stimuli that were next to each other, they got associated. He didn't look at the predictive component, but looked at the associative component. So that's one experiment from 1988, something like that. But maybe there is more. But yes, this is a very simple version. Now we can bring in the rat running at different speeds. This will still work, because this is a small delta t. We can bring in other things. This will become speed dependent. If the rat goes through very, very fast, very little happens. So we can start calculating all those interesting things. All right? So then we said, all right, let's look at the output. And when we look at the output, you get this. Notice that I told you, yes, please.
So there's the monosynaptic pathway and the trisynaptic pathway.
Yeah, I'm ignoring all that.
Okay.
No
One
trisynaptic.
is for generalization, the other one
I
is...
don't know that. I don't know that. Right now, I don't care. This is hard enough, so I'm just going to go along with this. Everything else, we are going to come later. All right? This becomes annoying. If I condition you to say I'm a neuroscientist, then it becomes annoying to say, what the hell, why is he making such simplifications? If one of you math guys start to talk about it, you say, yes, of course. So that's how we do things. So it's a occupational hazard of doing both. All right, so this is what happens. Now we are putting one more thing. We know that whenever the animal starts to run, this hippocampus develops these oscillations on their own as well. 8 Hertz oscillation, being bump, 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 bump. This is now the actual result of simulation with cosine tuned curves, random oscillations, which you can treat as noise. This is the activity pattern before. That's the activity pattern after, because the synaptics didn't happen. So from this, we can start becoming like Kepler. And we say, all right, what are the things that I should see in data if this was my model? So I should see the following things. First thing, when the rat is going from left to right, we are only treating one-dimensional thing. The first spike in the field as the rat's going through starts to appear earlier and earlier. That's my anticipation. The last spike should appear earlier, but by a smaller amount, because the amount of reduction in synaptic strength is less than the amount of potentiation. The net result of more excitation and inhibition is that the total activity should go up, and there should be symmetry breaking, and there should be this third moment. So the first moment of the distribution, total activity, first moment should increase, or 0th moment. The first moment is the center of mass should move backward. Second moment, standard deviation, it should get wider, but in an asymmetric way. And the third moment, with the shape of this, it should acquire a negative skew. How much we're going to look at it, that depends on the model parameter, but these 3 or 4 results are true no matter what you do with it. So if it's that, you can say, all right, if that's true, I can start looking at it, so I can summarize a whole bunch of predictions. I'll run out of time, so I won't go through that. So here's the data now. So the standard idea people had is that place fields are cosine tuned curves, which people saw in all parts of the brain. All neural tuning curves are cosine. So then I said, aha, if I want to look at the footprint of learning in any part of the brain, if that neuron's activity has anything to do with causal learning, its shape should be asymmetric. And this is the actual place field that I recorded. Notice its shape is asymmetric. And this was after a lot of learning, not in the first trial. So vast majority of this is just the third moment in dimensionless numbers. The skewness is about -1. Pretty strong effect. Majority of the neurons distribution have this kind of a ramp, not the other way, in this one-dimensional path. So this is after learning. We have not yet looked at how this changes with learning. So that's one solid prediction. So if you go to, now this hippocampus is highly plastic, so these things are changing very, very rapidly, so you can measure it. But let's say you want to understand, did the receptive fields in the primary visual cortex arise or are playing a role in some kind of sequence learning or causal learning? Those things all got wired up early in the development, so you can't see them evolve. Simply look at the shape of the receptive field. It's not all the story, but if it's skewed in this fashion and not the other way or mostly symmetric, that's a mark that this is some emergent feature of causal learning. That's number one. Number two is this prediction, the whole one. So I recorded one neuron here, but this is now not a model. This is one actual data. These are the spikes from one neuron. Somebody had a question about spikes. These are spikes from one neuron in trial number 1. These are spikes in the same neuron, and that's the thing that model can't tell you. How many trials will it need? Will it need 5 trials? Will it need 100 trials, 1,000 trials? We don't know. At least back in the day, we didn't know. It turns out this from here to here change happened in a matter of 2 minutes. The rat simply walked back and forth about 3 times, and already within 3 trials, you can see everything. And then this is a smooth version of it. So you just convolve these spikes with some kernel and you get a smooth function. So let's look at that, which we can compare with the model results. So you get this. So the rat went from left to right. This went back by a large amount. This went back by a smaller amount. Total firing rate went up, and the shape required this massive asymmetry. So everything that we stated worked for this one single neuron. Now we want to verify this. Say, one neuron can do anything. I can cherry pick the data and show you this and that. So you now say, okay, I got these few dimensionless numbers. Net firing rate should increase, center of mass backwards. This should be bigger than this, and third moment. So you verify the average across hundreds of neurons. You have to do a lot of dirty work. The rats, when they run around, they make a mess on the field. They pee and do other things. So you wipe it carefully and make sure all that doesn't happen. And this is the average thing. So the location of the first spike, so this is averaged across all the neurons. So with respect to the center, you can measure. So across the ensemble of about 180 neurons, the first spike shifted backwards by about 11 centimeters. Notice it was very fast. Happened within 2 trials, and then nothing much happened. First spike went back. by about 11 centimeters. The last spike in the field, meaning at the end, went by a smaller amount, about 6 centimeters over a slightly longer period. Interesting story. And the third moment of the distribution, because third moment is dimensionless number, I can average across all the neurons, was not significant before, and then it changed by a large amount, and then kind of became stable. So by doing this, and then there's a lot more. There's a big fat table with all the numbers about how much something changed and how you can match theory and experiment. So you can do that. There are many other controls I did. I have 2 more minutes left before I can finish up. So maybe I'll just pause here and see if there are any questions. Otherwise, I go. Yes, please.
You say that in terms of time, this centimeters anticipation, you said it's like 10 centimeters. How much time is it?
Very good question. It's about 250 milliseconds for the average speed at which the rat was running. On an average, about 250 centimeters, and that has a nice number to it. So what is 250 centimeter? You can measure in 250 millisecond. Is it just millisecond is what I count? It turns out, and that's the part that I was going to get into later on, the magic lies in this bumpiness, this theta rhythm. Notice this 1 bump, 2 bump, 3 bump, 4. It's a messy, but that rhythm is there. That rhythm has a period of about 125 milliseconds, pretty precise. That's called theta rhythm. So it's roughly 2 cycles, roughly jump 2 cycles. Yeah?
Is this a similar kind of mechanism to what would lead to edge detector cells?
Don't know that. Haven't done that.
But it seems like it could be if they're
we have, that's right. So
eyes along.
So I spent a fair amount of time trying to generalize this. I've written a paper about it called From Hippocampus to V1, how the structure of spatiotemporal receptive field can tell you what's going on. And I have looked at that in primary visual cortex if this is going on, which is hard, and I don't do experiments in that, don't have the ability to do those things. But there, the shift is by about 50 milliseconds in those oriented neurons, which are direction selective as well. In layer 4, there are lots of 5, I think, the direction selective neurons, they do this. But this is just my observation. It's not something that I have studied, nor anybody has studied and published. But this idea did catch on. Initially, it was too wild and so on, but then some people got excited. Instead of primary visual cortex, they took the brain of a xenopus. And that brain lives quite alive, and they say, if this is going on, it should work directly in the optic tectum. Don't have to go deep in the brain. They put the brain of this eye of this xenopus here and just took a bar of light and kept moving many, many times over in one direction. All of these things happen instantly. So that was perfectly controlled. That worked. And then people have done lots of experiments since then. So some people said, okay, but that was xenopus. What about the rat brain? So now novel technologies came up. They said, let's simply silence the connection, the plasticity from that CA3 to CA1. The CA3 was the input region, CA1 was the output. There were all the other things like trisynaptic circuit, this and that. They developed molecular genetics tools. And they said that simply silence the plasticity in CA3 to CA1, the activity is present, the rest of the circuit is unperturbed. If this theory is true, and if you simply remove the plasticity of an MDA receptor only between CA3 and CA1, all this should go away. All of this went away. So then people were skeptical, when you do physics kind of stuff, people say, yeah, but that's just because you messed with the circuit. So just 5 years ago, somebody found another trick. They said, what if I now boost the plasticity? Then it should get better. And the animal's ability to do causal learning should actually get better when you mess with some channel. So there are some weird channels called H current, which does some funny thing, but at the end of it, when you mess with that H current, the window gets wider. If the window gets wider of this Hebbian learning, then everything should happen better. Exactly as predicted. Things got better, and the animals actually got smarter. They solved the maze faster in a causal inference task. So there's a fair amount of evidence. I have written many papers about it since then. And so we wanted to then go for something really kind of out there to say, this is what one neuron does. But I want to go towards behavior. is this actually influencing the animal's behavioral decision? Because like one neuron out of this, and maybe on an average happens in CA1, but there are too many neurons. So depending on what the interest is, tomorrow or day after is my last lecture, I can cover one or the other. So I can either cover how to go from here to behavior, or I can talk about these declarative or other memories, or I can talk about how the entire deep network works, how it goes from visual cortex to hippocampus. There are 3 or 4 choices. Or a simple theory of what happens when the animal is not doing anything and how this circuit does some weird transformations. So different choices. You had a question.
Right. So, and then in your model simulation, it seems like the CA1 neurons have got a sense about like they say 3 neurons and then active and before me or after me. How could the neurons get a sense of the time?
That's right. Doesn't. But just statistically true. Neuron doesn't have any sense of time. Not now. You see, that's why this is important. The neuron has no idea what's going on, right? It's just true by construction. It's just true by construction that action potential, when it activates this neuron, these AMPA receptors are active for only 5 milliseconds. That's just a biological fact. The EPSP that generates the synaptic potential it generates is roughly half a millivolt. That's just a fact. That means that you need at least 10 or 20 neurons to be active simultaneously to cover the gap from minus 65 to minus 50 in order to generate a spike. Otherwise, the neuron is not even active. But once the neuron generates spike, the spike will go and muck with the NMDA receptor, remove the magnesium block, and it will start learning. So all this is true just because of the basic fact of biology that AMPA receptor time constant is roughly 5 milliseconds, and individual AMPA receptor mediated potential is half a millivolt, and the gap in order for neuron to generate a spike is 20 millivolt. Then this is more or less given. Now, it's not going to be perfect cosine shape, and the result will get somewhat tricky if the shapes start to be very weird and so on. That's another long story of what this can do. There's a question in the back and then you, sorry.
Nope, sorry. It is also a biological fact that after 80 minutes, people need to use the bathroom, get some coffee. So let's thank Mayank very much.